Hello everyone, my name is Rebecca and I'm going to present you a case study of a PBPK model development and validation and its use for evaluation of the impact of different dissolution rates on drug performance in vivo. The opinions expressed herein are solely mine and do not represent statements or opinions of Lick Pharmaceuticals, Sandoz Pharmaceuticals or Novartis. In this short presentation, I'm going to present you development and use of a PBPK model for a modified release capsule. I'm also going to emphasize best practices in development of PBPK models and present some challenges we face during modeling and opportunities I see for potential use of models in the future. First, let's focus on the case study. During drug product development, we had two modified release formulations with different in vitro and in vivo behavior. Only two formulations are not enough to establish in vitro and in vivo correlation according to the FDA regulatory guidance. We were thinking if it would be possible to establish regulatory acceptable in vitro and in vivo relationship using PBPK modeling. The purpose of this IVIVR would be to evaluate the impact of a change in dissolution profile on drug product behavior in vivo. The established dissolution specification for this modified release drug product consisted of 3 time points, the first one being 25 to 45% of drug dissolved in 2 hours. We wanted to evaluate the impact of changing the specification to not more than 45% of drug dissolved in 2 hours. The drug substance was a BCS class 1 drug. We used GastroPlus software for the development of a PBPK model. The dissolution method used as an input into the model consisted of 2 hours in 500 ml of 0.003% polysorbate 80, followed by 10 hours in 500 ml of phosphate buffer at pH 7.2. Based on some experimental work, this method was more biorelevant than first stage including acidic medium, where dissolution was too slow. The model was developed gradually. All available literature data on physicochemical properties were entered in the software. Then pharmacokinetic parameters were determined by fitting intravenous infusion profile in PK Plus model of GastroPlus. Since no data on permeability was found, effective permeability was set based on fitting simulated profiles for 3 strands of oral immediate release formulations to plasma concentration profiles observed in vivo. Finally, in vitro dissolution profiles were used to simulate 2 formulations of modified release capsules. As can be observed from figures, all simulated profiles matched profiles observed in vivo quite well. All calculated average prediction errors were below 10% and all individual prediction errors were below 15%. Thus, we considered our model as validated. In order to trust the model results, any model optimization needs to be justified. As mentioned before, effective permeability value was set based on immediate release products as there was no literature data available on, for example, KCO2 or animal permeability. There was also no plasma concentration after administration of oral solution. Part of model optimization was the increase of gastrointestinal transit time in cecum and colon in order to allow prolonged absorption of modified release capsules. The increase was still within physiological range observed in vivo. The last optimization was decreased default software permeability variability when simulating virtual trials, in order to capture variability in pharmacokinetic parameters observed in vivo. The developed model was used to simulate the plasma concentration profiles for a new formulation with a different dissolution rate, as shown in the figure with red line. This hypothetical formulation has 0% of drug dissolved in 2 hours. The simulated plasma concentration profile is similar to the test product with a slight delay in Tmax and similar Cmax and IUC. Also, virtual clinical trials showed bioequivalence of these two formulations. Based on these results, the change in dissolution specification was justified and also accepted by the regulatory agency. What best practices we usually follow when developing PBPK models? First is gradual development of models. For example, first developing a model for intravenous data to correctly capture distribution and elimination phase. Then use immediate release products to capture absorption phase, and lastly include modified release formulations. It is best to show the model predicts well different formulations, different strengths, and different physiological conditions, for instance fasted and fed state. With regards to permeability, if in vivo data after administration of oral solution are available, this could be used. Other possible cases include data after administration of immediate release oral formulations, data after direct administration to different parts of the gastrointestinal tract, data from in vitro tests such as Keiko2 cells or rat intestine. Thirdly, the biorelevant and biopredictive in vitro method is necessary to be used so that a model is able to discriminate between different formulations. Also, there should be at least two formulations with different in vitro and in vivo data to actually establish in vitro-in vivo relationship. What challenges we face during development of PBPK models? Sometimes we struggle with selection of appropriate model parameters. For example, when there are multiple significantly different data for compound-specific parameters, one has to decide which to use, make some in-house measurements or parameter sensitivity analysis. Also, with regards to population physiological parameters, due to high variability in the gastrointestinal tract, there are still questions which mean values and what variability should be used. With regards to in vitro inputs for the models, it is sometimes quite challenging to find biorelevant and biopredictive dissolution methods, especially for non-oral dosage forms. Also, permeability values and their variability for both oral and non-oral dosage forms are not easily found and justified. When performing model validation, there sometimes isn't enough in vitro and in vivo data to properly develop and validate the model, for example, no non-bioequivalent data. On other occasions, there could be a lot of data, however, there could be significant interstudy differences. This complicates the validation of the model, especially when trying to reach 10 or 15% prediction error. There remains this question of what prediction or fold error is appropriate for a specific model. For example, if the intention of the model is to predict bioequivalence of the formulations, in our opinion, it is more important that the model adequately describes the ratio between the test and reference formulation, and it is not so important how it describes the absolute values of Cmax and AUC observed in the in vivo study. The challenges we face during development of PBPK models are actually quite similar to the challenges using IVIVC reported in a survey from 2017. In the survey, different parties emphasized the challenge with lack of appropriate clinical data to develop IVIVC, regulatory uncertainty, lack of time, resources, and skills, the problems with IVIVC validation, lack of appropriate dissolution data, and no difference in the in vitro release of different formulations. However, IVIVC was said to be frequently used as a supporting tool in drug product development, as it provides better mechanistic understanding of the formulations. We think the same is true for PBPK models. I believe in the future we will find a way to solve all the challenges. So what are the opportunities that PBPK model will then be useful for? There is a possibility to waive fed bioequivalence studies required by some regulatory agencies. There have also been ideas to justify bio-waiver for BCS class 3 drugs with Q1 and Q2 differences. The models could also be used to show bioequivalence for different non-oral drug products, avoiding clinical endpoint studies. PBPK models are also used for justifying different scale-up and post-approval changes, such as the case I presented. In the end, I would like to thank all my colleagues at Sandoz, both in SDC Slovenia and globally, for many scientific discussions about modeling. Here are the references used during the presentation. Thank you for listening. Please provide any questions in the Q&A box. I will also join the panel discussion.